Ladies and gentlemen, welcome to the Royal Society, especially if this is your first visit to the Society, and particularly this evening to this year's Francis Crick Award Lecture. I'm Jean Thomas, and I'm the Biological Secretary of the Royal Society, among other things, which is why I'm chairing this lecture. And before we get into it, could I ask you please to switch your mobile phones off, right off, because the lecture is being streamed and webcast, and we don't want nasty noises getting in the way. And if you've had a chance to watch the screen, as we've been sitting there, you'll see that this lecture happens in the Royal Society's 350th anniversary year, and this has been marked with a number of events. It's almost over at the end of November, but the highlights of the year have been the Summer Science Festival on the South Bank, publication of a book that was edited and introduced by Bill Bryson, the Capital Science Programme in partnership with museums and galleries across London, and the Local Heroes Programme, where people are celebrated on their own patch, as it were, right across the UK. And the latter two are still going, so you have time, if you haven't done anything yet, to catch up with some of these events before November the 30th. So we've been celebrating hard all the year, and as I say, it's about to come to an end on Anniversary Day. So to get to this evening's lecture, the Francis Crick Lecture was established in 2003 from an endowment by Dr. Sidney Brenner, joint winner of the 2002 Nobel Prize in Physiology or Medicine. The prize lecture is delivered annually in any field of biological sciences, with preference given to the general areas in which Francis Crick himself worked, namely genetics, molecular biology, and neurobiology, and fundamental theoretical work, which is the hallmark of Crick's own science, will also be given preference. And importantly, the lectureship is aimed at younger scientists at an early stage of their career. So the Francis Crick lecturer for this year is Professor Gilean McVean. Gilean McVean was an undergraduate in Oxford, reading biological sciences between 1991 and 1994. There followed 3 years of graduate work in Cambridge in the genetics department, which was a very productive period by the look of things. Several papers emerged, and he worked on the conflict theory of genomic imprinting and the evolution of mutation rates. Next 3 years, this brings us to 1997-2000, postdocing in Edinburgh with Brian and Deborah Charlesworth, and working on theoretical population genetics, particular modelling the effect of natural selection on a genomic scale. So you see the theme emerging here right from the start. In 2000, Gilean McVean was awarded a Royal Society University Research Fellowship, one of our prestigious career awards, to work in the Department of Statistics in Oxford. In 2004, in that department, he became a university lecturer in mathematical genetics, where he was involved in a number of high-profile genomic studies. But in 2007, he became professor of statistical genetics. I think I've got that right, hopefully. This whole crew from Oxford in the front here. Recently, he became co-chair of the analysis group of the 1000 Genomes project, which you may have heard about, a large international project aimed at sequencing 1,000 human genomes for comparative purposes. So Professor McVean was awarded the Francis Crick Prize Lecture for his major contributions to the field of population and statistical genetics. So without further ado, I introduce our speaker this evening, Professor Gilean McVean, who will speak on our genomes, our history.
Thank you very much, and it's, of course, a huge honour to be asked to give this lecture in memory or in honour of the great Francis Crick. My students always tell me that I don't dress very well, I'm somewhat dishevelled, so I'd put that picture of Francis up there to just show that even the greatest scientists can sometimes look a bit dishevelled, although I admit he is wearing a tie, which I haven't quite managed to do. So Francis Crick was, of course, the godfather in many ways of the current biology, current efforts to try and understand how you turn a genome, this incredibly long sequence of DNA that sits in all our cells, into the sort of the fully functional, interesting entity that is the person sitting next to you. So in many ways, it's a great time to be talking about DNA, and in particular the human genome, because this year marks roughly the 10th anniversary of the publication of the first draft sequence of the human genome. So in 2001, the big splash, scientists got very excited because we had the first single view of what the human genome looked like in its entirety, the 3 billion letters that goes up to making your genome. What's happened in those 10 years is that scientists from all over the world have been busy trying to map on all the bits of biology that go into turning this sort of raw sequence, just A's, C's, G's, and T's, into the functional unit, which is the cell. And so whether that's mapping where the genes are, or how those genes are turned on, or how the various bits of, or how the DNA is regulated, lots and lots of different information have been mapped onto that one entity, that human genome. But of course, there is actually no such thing as the human genome. We all have completely unique genomes, and it's the particular combination of genetic variants that we have that goes such a long way to making us, the person that we are. Of course, there are other influences, but if you just look at the people in your family, you can see that there are all sorts of peculiarities that you share, and it's because of the DNA. And so what that means is that to really understand how the DNA turns into the individual and how differences between genomes can make people look and behave differently, we need to understand the genetic variation. And so this year, 10 years after the publication of the first draft, we have the publication of the first findings of this project that Jean mentioned, the 1,000 Genomes Project, which I'm very delighted to be a part of, which is incredibly exciting. And what it's aiming to do is to chart the complete genome sequence, effectively, of initially just a couple of thousand individuals or so, but in order to try and map out the genetic differences that we have, and ultimately, of course, to try and understand how those differences influence what I end up being. So for scientists, it was an exciting time. This was published only a couple of weeks ago. It was a good excuse for a party, although if you're The Guardian, it was a good excuse to print a picture of a woman in a leopard skin bikini. So what have we learned about the genome from looking at genetic variation? Well, just to back up a bit, as I said, the human genome is about 3 billion base pairs long. You've got 2 of those in every cell, one from your mum, one from your dad. So you've got about 6 billion nucleotides. That's 1 for every person on the planet. If you stack all the nucleotides together, you get something that's about 2 meters long, but by some complete miracle of engineering, that's managed to be compressed into something only a few microns long in every cell, and you have this basically in every cell in your body. What do genetic differences look like? Well, let's zoom right back down to the level of the letters in the DNA that sort of lie in the center of this double helix. So here's just a little bit of the human genome. This is only a couple of thousand or so letters. And to give you a sense of the scale in which our genomes differ from each other, in there are a couple of red letters that sort of give you a sense of the average level of difference between, say, my genome and your genome. And here there are just 2. So there's about 1 difference between my genome and your genome every 1,000 or so letters. Now, that doesn't sound very much. We are incredibly similar to each other at the DNA level. But of course, remember that our genome is very big, 3 billion letters, so that's 3 million or so differences between my genome and your genome. And so there's a lot of potential in that to create the kind of physiological or downstream physical differences that people clearly show from each other. So it's a very simplified view, just the odd letter that differs from each other. Of course, biology is more complicated than that, and in fact, there are many different ways in which our genomes differ from each other. You can have, as I said, these single letter changes, happen every 1,000 or so. You can have little insertions or deletions of groups of letters. Here, this person has 3 letters that person didn't have. There are some regions where you find particular units or little groups of letters, little words, which are repeated different numbers of times in different individuals. So here's this group of 3 letters, TCA, that this person has twice and this person has 4 times. These are called microsatellites. Going up a level, you find things that are sort of repeats, and again, people vary in the number and type of these repeats. So here that's just 3, but here we've got repeats that vary also a little bit in their sequence, and these things are called mini-satellites. And those are the things that were initially used in DNA fingerprinting because they're very variable. And we'll come back to talk about those later on, so just bear that in mind. But then going up a scale, there are bigger and bigger differences between our genomes. There are some of our genes sit in these massive arrays, tandemly duplicated arrays, and there are big differences, subtle or substantial differences between individuals in the number and the type that people have of these. And indeed, you can have even larger things. So, for example, the Y chromosome, which is what goes about making men, real men. You can actually have quite large deletions on this Y chromosome and not have a huge impact on, say, fertility or whether you're going to end up a male or not. So our genome is sort of peppered with these variants of different forms, at different scales, of different complexities. And by sequencing this 1,000 Genomes Project, what we're doing is trying to work out sort of the map of where these things are. Now, so that's one way in which sort of there's granularity to the type of variation we have. But when you start looking at genetic variation across the genome, you start seeing interesting patterns. So this is just a map of genetic variation across the human genome. We've got 22 autosomes and a pair of sex chromosomes. This is just the autosomes, the things that aren't the X and the Y. And it's just colored by how much variation there is. So red means lots, blue means little. And what you can see immediately is that there's structure to this. It's not a sort of a uniform color. Indeed, there are some regions that have a lot of variation. It turns out that this particular region is the part that's really important for whether or not you match a patient can accept a transplant from another person. It's the thing called the MHC. And there are some other regions which have very little diversity. And this is a region, a gene-dense region. We don't actually really know its function. It looks kind of interesting, something to look at. So what you see is that there's sort of patterns in this variation. And so what we'd like to do is to understand these patterns. And so we need to be able to ask the question of where does this genetic variation come from? Why do we see these differences between genomes? Now, of course, there's a very simple answer to that. And that answer is just that there's mutation. When DNA copies, it changes. It makes mistakes. There are chemicals out there in the environment that change it. It just has random shifts. So these mutations are happening to our genomes. But actually, they're happening at a pretty low rate. Our machinery for copying DNA is fantastically good. It has remarkable fidelity, so that in taking my parents' genome and sort of putting it into me, the number of errors that were made in that process is about 30 or so in 3 billion. It's phenomenally good at recording the information. And so, of course, if you've got 3 million differences between me and you, that's not just a result of mutations that came from my parents. There's a lot more to explain. So where is that coming from? So let's try and do a little experiment. There's me on the left. And what I'm going to do is to think of my DNA, and I'm going to follow it backwards in time. I'm going to start with it in me. I can look back, and I'm going to just take one of my chromosomes, and that will have come from one of my parents. Suppose it came from my father. That might have come from his father, and so on. And so this chromosome that I've got essentially has its own little journey backwards in time through history. And it keeps on going back and back and back. And in fact, of course, it just goes back to the origin of life itself. You could ultimately trace this bit of DNA all the way back to whatever it is, 3 billion years or so that life has been around, although be somewhat fragmented by that time. But actually what I'm interested in is the differences between my genome and your genome. And so I'm going to take you over here, and I'm going to take your chromosome, and I'm going to follow that backwards through time. What's going to happen? Well, of course, you had some parents. Your chromosome came from one of those parents, and so on and so on and so on. But at some point in the past, I can guarantee that we will have shared some ancestor. I might have had to go back 500,000 years. I might have to go back 1 million years. I might even go back only 2 generations. Who knows? But there is some shared ancestor. And that's true for me and the person in the front row, and it's true for me and the person in the last row. And so the differences between my genome and your genome, you can think of in terms of where changes have occurred in this long history that represents our sort of shared genealogy. So the red blob there is meant to represent a chromosome that's been transmitted back down through the generations into me. It is going through there. And then I can trace that chromosome again down through the lineage to you, but maybe at some point that chromosome had a mutation. Maybe when that bit of DNA was being copied to make that particular child, there was a little error. And so something crept in, represented by this big flash of lightning here. And then what's happened is that changed that DNA a bit. Maybe it's one of these point mutations, or maybe it's a big inversion or something. And then what's happened is that that bit of DNA has been copied down to you through the generations. And so the differences between me and you are really the result of mutations occurring somewhere in our ancestral history. If you like, they're the mistakes made by our ancestors. Now, that's a very simple sort of cartoon. That's just me and you. What if I take more people? What if I take everybody in this room? Well, what happens if I take more and more people? The same is true. We have this shared ancestry, this shared genealogy. And so for any 2 people, there is some common ancestor at any one point on the genome. So imagine that the 2 people I was talking about was the person there and the person there, and that's the particular common ancestor they had. But I take another person, I can find the common ancestor of that person and that. Take someone over here, I have to go further back. And so if these are our sort of genomes down at the bottom, and the blobs represent the mutations that make me different from you, different from the person over there, then you can think of these differences as occurring on this tree. There is a tree that essentially tells us how we are all related to each other. It's different in different parts of our genome. There's some complexity there. It's actually more of a sort of complex graph structure. But you can represent at any one point in our genome our history by this kind of tree. And so if you can think about when mutations have occurred and how that relates to human history, it's kind of interesting, because in order to find this sort of ultimate common ancestor back here, modern humans split from chimpanzees about 5 million years ago or so. You have to go back maybe up to about 1 million years to find this common ancestor of all the people in this room, essentially. Now, think about that. Modern humans arose about 100,000 years ago. So actually, the vast majority of this tree isn't modern humans. It's when we were something more archaic. Actually, quite a lot of this is what's called, is essentially variation. These mutations have occurred when we weren't Homo sapiens, but we were actually Homo erectus or something like that. And so these mutations clearly are, in some sense, a record of human history over the last 1 million years or so. The differences that we see between ourselves at the DNA level are the result of these mutations which might have occurred up to 1 million or so years ago. And so more generally, you can think of genetic variation. The difference is, if you want to try and understand genetic variation, you can think of it as being influenced by all sorts of processes that have acted over human history. Obviously, there's this process of mutation, the actual way in which new mutations, new changes to this DNA happen. There are these processes like recombination, which I'll talk about again in a second, which shuffle up genetic variation. There are processes, Darwinian selection, which have shaped the fate of mutations. A mutation appeared, for example, that about 10,000 years ago or so, that made people living in North Europe able to digest milk into adulthood. And that mutation was hugely beneficial, and because it was hugely beneficial, it sort of did very well, and now almost all Europeans share this mutation. So there's selection going on. There's population history, the way in which different peoples have moved over the surface of the globe over evolutionary time. And of course, there's chance. And chance plays a big role in everything in biology. And so what we can imagine is that we can easily measure genetic variation, relatively easily, and what we'd like to do then is to turn the sort of the study of genetic variation into an understanding of these underlying processes. Our genomes are a coded version of the forces that have shaped human evolution. Can we just take these patterns of genetic variation and tell us about those processes? And that's really what my research is about. And so to try and give you a sense of what has happened in my research in the last 5-10 years or so, what I thought I'd do is to pick out a couple of stories which relate to how we think about genetic variation and these underlying processes, to give you a sense of the sort of the complex biology that you can learn simply by studying the genetic differences between individuals or between DNA molecules. Now, the first story I want to tell you about is I've called the mutating genome, and it's about the processes that actually go into changing DNA from one generation to the next. Now, the secret in science is to have good collaborators, and I've been incredibly fortunate over the last years in working with fantastic people. And the two heroes of this story are Andrew Wilkie and Gorielli, who I believe are somewhere in the audience, so if they're sitting next to you, give them a cheer. And this story is essentially about the way in which mutation works. Now, as I said earlier, our genomes are unique. We all have an individual genome. Actually, that's not true. We probably have 10 to the 14 different genomes or something. Every cell in our genome has a unique genome. That's a pretty scary thought. Because every time a cell copies its DNA, or just by standing still, DNA is changing. And it's changing for a whole bunch of reasons. You can have spontaneous chemical changes. There are particular sort of types of DNA change which are happening thousands of times every day in each of our cells. Fortunately, we have a machinery that goes around correcting this. But these things are happening very frequently. You might come into contact with environmental mutagens, things like UV light, for example, things that actually go out there and damage DNA. Many errors in DNA are probably made when we try and copy it from one cell to another. And you can have things like the copying machinery can just slip a bit and sort of leave in or take out a particular bit. It can sometimes stall and fall off, and it does all sorts of complex things to try and recover, which can have some quite nasty consequences. And then there is a copying process. Fortunately, it has a proofreading stage, so it's incredibly accurate, but it's not completely faithful. Every now and again, it does make errors. So there's lots of things happening to our genome, or to our genomes, and they're changing it all the time. And this particular story is about the perils of aging, and in particular about the perils of marrying older men. So it's sort of long been known that marrying old men is a bad idea for many reasons, unless they're fantastically rich. And one of those reasons is that there are a bunch of diseases out there which are genetic diseases. That means they're caused by particular changes in particular genes. And if you look at, so these mutations nearly always happen de novo every generation. So you don't pass these mutations on. If you get this mutation, you're unlikely to have children. And so these mutations are things that are happening recurrently every generation, but every time it's essentially new. Now, these are diseases hopefully none of you have had or had any contact with. They're typically fairly rare. Some of the more common ones is achondroplasia, the most common form of dwarfism, for example. There's also neurofibromatosis, which creates little lumps all over you. And what these charts show you is how, as a function of paternal age, the incidence of these diseases increases. And what you can see here's one called Apert syndrome, which increases pretty sort of exponentially with paternal age. That means, so your chances of having an Apert mutation in your offspring increases, I think there's a 60 up there. It's either a 6 or a 60. Something like either 6 or 60, I can't read it, fold over this sort of age span of paternal age. So what's going on? Why is it bad to be an old dad? Well, I'll get to that in a second, but one thing that you need to understand, or one bit of information, is that, as I said, these are diseases caused by mutations, in fact, particular mutations in particular genes, and all the genes underlying these particular syndromes have been identified. And just to give you a sense, these are not random genes. There are particular genes that really come out of these. And in particular, there's a group of genes called the FGFRs. I'm not going to tell you what that's about. But these are genes involved in cell proliferation, which is an important thing. I shall come back to that. So this Pfeiffer syndrome, that's caused by mutations in FGFR1. These three syndromes are all caused by mutations in a related protein called FGFR2. And this syndrome is caused by mutations in another one called FGFR3. So there's three proteins, and all of these paternal age syndromes are caused by mutations in these particular proteins or genes. Now, there's another one up there, neurofibromatosis, that I mentioned earlier. I'm going to come back to that one later, so just remember that name for a bit. So what's going on? Why is being an old dad bad? Well, the idea that's been around for a long time is essentially that the problem with being an old dad is that as you get older, mutations are accumulating in your germ cells, in your sperm cells, essentially, because they carry on replicating. So as you get older, the cells are dividing and dividing and dividing. Every time they make a few errors. And so those errors accumulate. And so by the time you're an old dad, you've had a lot more rounds of cell division. And so you're more likely to have one of these nasty mutations. So by the age of puberty, there are about 30 cell divisions that are a male sort of germ cell. These are the cells which are going to make sperm. They've been through about 30 cell divisions. And then after puberty, you have about 23 or so divisions per year. So by age 25, you've been through about a bit over 300 cell divisions. And by age 70, you've been through getting on for 1,400. So there's a threefold difference in the number of cell divisions that you've been through. And that, you'd think, would lead to a threefold, at least, increase in the rate at which mutations are going to happen. So, of course, a mutation that occurs at some point in here is going to essentially be carried on through subsequent generations in this population of cells. And so what Andrew and Anne were interested in doing was trying to understand the exact process by which this was happening. And they kind of smelled a rat, which was that, as I said, these are not random genes that these mutations are in. These are particular types of genes which are involved in cell proliferation. So what they wanted to do was to be able to actually measure the frequency at which these mutations were happening in sperms. They wanted to take men, get their sperm. It's remarkably hard to get consent for that. You'd be surprised. And actually measure the frequency at which these mutations were happening in this sperm. And rather than trying to solve the whole thing, they're just focusing on APERT syndrome, or I was involved in this project, obviously, focusing on APERT syndrome, and in particular, a particular mutation that caused APERT syndrome, which is one of the most common disease-causing mutations in the human genome. So about 1 in 70,000 or so live births will have this particular mutation. Now, the background rate of mutation is about 1 in 10 to 100 million. So this is massively increased over the sort of background rate of mutation. So how do you go about measuring mutation? Obviously, what you'd really like to do is to take every individual sperm, look at its genome, ask what letter it's got there, and sort of tally that up. Now, that's actually hard to do. People are working on single-cell sequencing at the moment, but it's not mature. And so currently, the best way of trying to estimate these frequencies is to actually look at a population of sperm and say, what's the frequency of this particular mutation in that population? So this is my cartoon trying to represent a whole bunch of sperm. And the blobs in here are the DNA. And some sperm, I should say, carry this particular mutation. And so what we want to do is to measure the frequency of that. How do you do it? Well, the first thing you do is you get the DNA out of the sperm. You then add in a sort of a known concentration of something else that you're going to measure this DNA against. You get rid of all the DNA that isn't mutated by adding a Pac-Man. So that gets rid of all the what's called the wild-type DNA. And so you're left with a known concentration of something that you've stuck in and these other mutations. And so you can essentially just sequence this mixture, and that allows you to work out the frequency of these mutations. Now, it sounds easy, but of course it's tricky. And my role in this project was essentially to devise a way of doing that robustly and cause them huge amounts of pain by making them do lots and lots of experiments to show that it's really robust. But it is remarkable. You can actually measure the frequency of mutations down to roughly at least 1 in 100,000 and close to 1 in a million. And I don't want to go through the details, but the bottom line is you take a bunch of men of different ages, you measure the amount of this particular mutation that they've got, you find a sort of spread of mutation frequencies, so a pretty big spread. But if you sort of put that all together, then it recovers the paternal age effect that was seen previously, that's seen from the epidemiological studies. And so that's good. It says that the frequency of these mutations in the sperm is reflecting what we know from the rate at which people get this disease as a function of their paternal age. So that's reassuring. It's just saying there's nothing else going on. It's just what's happening in sperm. But it doesn't tell us about mechanism. Because remember, these mutations are happening really much more common than you would expect simply on the basis of sort of simple DNA errors. So what's going on? Why are they mutating so fast? Is it just that the DNA sequence is really hard to replicate? Is there some hypermutability about the context in which that sequence lies? What's going on? Well, to cut a long story short, clearly what we found that's going on is a process that mimics natural selection. So what's going on, essentially, is that there are mutations occurring in these testes, in your testes. In fact, every male's testes in this room will have these mutations. And these mutations essentially create little cancer-like growths. I mean, they don't turn into cancers. They don't have any pathogenesis in that sense. But they create little sort of growths of cells. And they're growths, and because they're growths, they leave more and more descendants. It's just a process of natural selection. But rather than selection happening in a species as a whole, it's selection happening in your very own testis. How do we know that? Well, the way in which we knew that was by comparing a mutation that didn't have this particular phenotype, it's a different mutation, this. And if, as I said, we have 2 chromosomes, one from mum, one from dad. And if mutations were just happening very frequently, then if we looked at where the mutations were occurring, they'd happen essentially equally on the chromosomes from mum and the chromosomes from dad. So they'd be equally split over these 2. But actually what we found was that the vast majority of these mutations were sitting on a single background, which indicated that they'd essentially come from a single origin. There weren't many independent origins of these mutations, so they're occurring infrequently, but when they occur, they have a selective advantage. And that is a hypothesis that has since been corroborated by lots of other work. And so here's the idea. There's my testis. Well, hopefully not my testis. It's not as big as that. I've got these stem cells in there which are going to ultimately produce the sperm. So what happens is these cells divide, and those blue ones are going to go off to differentiate to produce sperm. And that goes on normally. But at some point, there's a mutation. And what happens is that causes that particular group to proliferate. And so locally, you get this sort of growth of this particular cell type. And ultimately, that can lead to you having a frequency of this particular mutation in your sperm of up to sort of 1 in 10,000 or so, maybe even higher. And so these, as I said, these don't create any pathogenic cancers, but they're essentially happening within all our testes. So that's kind of interesting, but is it a one-off? Is this just what's happening in Apert syndrome, or is it a general story? And the answer is that it does seem to be general, at least for these diseases that show strong paternal age effects. And additional work has shown that essentially the same thing is happening for the FGFR3 mutations that cause achondroplasia. You find common selfish mutations both in the germline and also in actual tumors. So some of these mutations are going to actually crop up in particular tumors as well. And essentially what's happening, these are all mutations which activate proliferation or cell growth pathways. So all these strong paternal age effects, it's not a result of this cell division sort of errors accumulating. It's a result of a natural selection-like process going on within the germline. And there's a chance that we might just be looking at the tip of the iceberg there. Maybe this is a common way in which sort of activating mutations can become common in a population. We don't know. That's an interesting hypothesis that we're going to go on to look at. Okay, so that's the end of my first story, and it's just to give you a sense of how complicated sort of mutation can be when you sort of drill down to it at the molecular level. The next story I want to tell you about actually turns out to have quite a lot to do with recombination too. Sorry, with mutation too. But it is ostensibly about something different. It's about recombination. So for those that don't know, recombination is a process you've all been through many times. Oh, yes, I should say the heroes of this story are, again, in the audience, so give them a cheer, Simon Myers and Peter Donnelly. So recombination is the process that goes on when I take 2 of my chromosomes and pass them on to my children. So here I've got 2 copies of each chromosome, one from my mother. If she looks familiar, she's sitting next to you. One from my father, likewise. I thought I should embarrass them. And what happens when I have children, then you don't just take one of these chromosomes and pass it on intact. Rather, you make a mosaic of them and pass them on. And that's why we kind of look like a mosaic of our parents. It's because of this sort of blending process. So it's a very fundamental process. Basically, all forms of life go through some kind of process like that results in genetic mixing. So it's totally fundamental. But until recently, we actually knew very little about where it happened in the human genome or why it happened in particular places. And so if you sort of go back a few years now, I got interested in trying to understand where this recombination process was happening in the human genome. And it turns out that although sort of traditional ways of learning about this process would involve either sort of studying inheritance of genetic markers through pedigrees or also doing similar sperm experiments to the type that was done with Andrew, actually there's another source of information about recombination, which is simply to study the genetic variation of unrelated people. I'm not going to tell you why you can learn about that, but broadly you can think of genetic variation recombination shuffles genetic variation. So to find out where recombinations happened, you just look at where variation has been shuffled. It's pretty simple. It's kind of simple, at least in theory. Being a statistician, I like to make the case that you have to use sophisticated statistical machinery to try and learn about where recombination is actually happening, but you can actually also just look at pictures and point at it. And so going back a few years now, we're interested in trying to map out where recombination happens in the genome. And what was important about this was that we could do this at a genome-wide scale. We could take the whole genome, we could look at where genetic variation was. We happily managed to coincide with some big projects that were happening at the time to map common variation across the human genome. And so from these maps, and this is just a sort of image of the raw data, we could actually take that, put it through some kind of statistical machinery and learn about where this process of recombination happened. Where is this mosaic crossing over actually going to happen? And when you do that, you find actually a really rather intriguing picture. This is, if you just look at the black lines here, this is one of our chromosomes. It's chromosome 12. I haven't picked it for any special reason. The black line is the recombination rate along the chromosome. So that means at this particular point on the chromosome, how frequently do you get these switching over mosaic kind of events? And what you can see is that this recombination profile goes up and down a lot. Okay? So it's essentially a landscape dominated by very short, very high spikes separated by regions of not much. Okay, and so we were essentially the first to chart. We were the first to chart this on the genomic scale in humans, and so it sort of led to an understanding of this recombination. Most recombination is clustered into about 5%, maybe 10% of the genome in short windows, about 1,000 base pairs long or so, in which recombination can be elevated by hundreds or even thousands of times. Okay, so this mosaic process is not smooth and happening evenly across the genome. It's happening in particular places in a landscape like this. Okay, so that's interesting. It tells us there's something going on there. Recombination is not a random process. What is driving this incredible heterogeneity? Why should it all occur in these spikes and not elsewhere? And so what we embarked on a sort of a process of discovery using essentially largely statistical techniques that just try and take the human genome and that map of recombination and find out, are there particular DNA words, for example, that can localize, tell you where this recombination is going to happen. And to cut a long detective story short, what we, or I should rather say Simon, identified was a little sequence motif. Here's just 13 DNA letters, very short little thing. And this plot is meant to give you a sense of what you would look for in the DNA in order to say, here's going to be one of these places where recombination spikes. So you'd look for a couple of C's, then you wouldn't care what that next letter is. A couple more C's, don't really care. Then a T, probably, don't really care, don't really care. C, don't particularly care, C. So it's kind of a fairly simple motif. There are obviously lots of these in the genome, but pretty much you need one of those in order to have a hotspot. That may not be quite true. And so, there's a little DNA letter, DNA word, and that word we find is sort of very strongly enriched in these hotspots. So it's interesting, but what does that tell us about function? Well, not very much. But there must be some way in which this little DNA word is recruiting recombination. So what's happening there? Now, unfortunately, I don't have time to go through the whole story, but it is a beautiful piece of detective work, putting together information from all sorts of different places. This chart just shows you how sort of powerful this little DNA word is. So if you might know that the human genome is stuffed full of repeat DNA. One of those repeats happens to be called THE, one element. Don't worry about what that means. And if you don't have this DNA motif, you do tend to get a little bit of recombination. So the idea is the center here is your average, the middle of your average element, and in blue is the average rate across all of these elements, and in red is what you get if you stick one of these motifs on it. This is a kind of sort of natural experiment, and you can see this motif is very good at sort of causing these spikes in recombination. So it's real proof that it is actually something that's driving recombination to these particular parts of the genome. So as I said, I don't have time to go through the whole detective story. It is really remarkable. But what we managed to find out was that the way in which this little DNA word is generating these hotspots is by being bound by a particular protein, something called PRDM9. Not a very exciting name, but that's what it's being bound by. And PRDM9 is something called a zinc finger protein. And what that means is it's got a bunch of loops which are stabilized by zinc ions. And those loops, these protein loops, interact with DNA, and they can interact in a sequence-specific manner. Okay, so in particular, there's this group of 4 amino acids, those are the sort of the chemicals in proteins, which binds specifically to groups of 3 overlapping bases. And you can make sort of good predictions about what they should bind to. So this is just a cartoon representing the zinc fingers in this protein, and this is representing what it should bind to. You can see there's something that looks remarkably like the motif that we've got up there. So this is essentially a purely statistical approach to identifying it, but these results have been confirmed by direct experimental work, PRDM9 is indeed the protein that picks out these little motifs and binds to it, and ultimately causes recombination. Okay, so that's interesting, but so what? We've identified a protein that generates recombination hotspots. Why should I care about that at all? Well, what I can say without any hyperbole at all is that it turns out that PRDM9 is the most exciting gene in the human genome. And it's the most exciting gene for a whole bunch of different reasons. So first of all, we think of humans and chimpanzees as pretty closely related to each other. If you take the DNA sequence, on average, they're about 1% different from each other, which means that two proteins, if you take a human protein and its equivalent in chimpanzee, there's typically at most one amino acid difference between these two. Okay? If you take the human and chimpanzee PRDM9 sequences, in particular you focus on this zinc finger region that's going to actively bind the DNA, you find that they're not just slightly different, they are completely different. This is the chimp sequence here, this is the human. Apart from this first zinc finger over here, they're basically completely different. That's very, very unusual. This is perhaps the most diverged gene between humans and chimpanzees. And furthermore, work by another group showed that this isn't happening by chance. This change is really being driven by some kind of adaptive evolution. So there's a Darwinian process driving divergence of these genes, not just between humans and chimpanzees, which is certainly true, but actually across all mammals, basically. So this is a protein that's driving at orders of magnitude, that's evolving at orders of magnitude faster than your typical protein in the genome. I'll come back to why that might be later if I have time. So it's massively different, and not surprisingly, that has consequences, and it turns out that chimpanzees have completely different sets of hotspots, not surprisingly. So we think of, again, as humans and chimpanzees being almost identical at the cellular level in terms of the sort of molecular processes. This is completely different. So something has completely switched between humans and chimpanzees in terms of where their recombination hotspots are occurring. What this chart shows you is, so the red, black, and green lines are sort of average recombination rate around your average hotspot in humans. You can see it's all nicely peaked. Blue is the chimp, and you can see that completely flatlines. There's essentially no sharing of these recombination hotspots between humans and chimpanzees. And so it's completely different between humans and chimpanzees. What's more is it turns out, again, this is work by another group, that this gene is actually also quite variable within humans. Not so much in Europe, but if you go to Africa, there are really quite different versions of this protein around, and that actually also has implications for which hotspots that people use. So people with different versions of this protein use different sets of these recombination hotspots. So that's kind of interesting, but again, what are the consequences of this? Well, one of the consequences goes back to mutation, and this idea that our genome is not a static object. Now, as I said, there are lots of different ways in which mutation happens. One of the most sort of striking and dramatic ways in which our genomes can mutate is through a process of recombination, but where recombination occurs between the wrong bits of our genome. So in this process of recombination, we have two chromosomes. They line up with each other, and recombination happens between them. And so they line up nicely. So they find out where they're sort of homologous to each other. But our genome is full of repeat DNA. It's absolutely stuffed full of it. And one rather unpleasant consequence of that is that occasionally the wrong bits of DNA can misalign. In particular, if there's a sort of a piece of repeat DNA that's meant to be represented by this red blob here, which is pretty like another bit found somewhere else in the genome, this orange bit over here, then they can end up lining up with each other. And you can have crossover between those two. Okay, so you can get crossover, and that can do one of two things. That can sort of just introduce a bit of the red repeat into the orange, and you go off and These are okay, so that's not too problematic. But much more problematic is that you can end up, if this recombination event is resolved in a different way, you can end up either getting rid of a whole chunk of DNA or duplicating it. And so there are a whole bunch of syndromes which are caused by this kind of mutational process. Now, these, again, are quite rare syndromes. Hopefully no one in the audience has these, but they can affect up to 1 in a few thousand individuals. These are the most common mutations driven by this process of what's called non-allelic homologous recombination. They've got some rather sort of nice names. But here's one that you've seen before, neurofibromatosis. This is the gene that also shows a paternal age effect. Actually, it turns out that paternal age effect is almost certainly driven by point mutations, so not these illegitimate recombination processes. And so there's a paternal age effect from the kind of selection process in the testes driving NF1, but there's also this non-allelic recombination process, which is actually largely driven in females. So if you put those together, it turns out that this poor old gene is probably the gene in our genome that's hit most by mutations of all the genes at all. And so maybe 1 in 10,000 or so of our children or individuals have a mutation in this gene, either through this paternal age effect process or through this non-allelic homologous recombination process. It just really didn't luck out, this gene. Okay, so why am I going on about this? Well, these are all these gene disorders caused by this illegitimate recombination. It turns out that not only is this motif that PRDM9 binds to basically lie at the center of all of the individual places where this illegitimate recombination is happening. So what that's saying is that PRDM9 binding to this particular DNA motif is driving this mutational process. That's pretty bad news. Recent work by Alec Jeffries has actually showed that this variation that individuals have in their PRDM9 actually leads to differences in the rate at which these mutations occur. And so what that means, if you put this all together, is that because individuals differ in their PRDM9 and humans and chimpanzees differ in their PRDM9, then different individuals are going to be exposed to different types of these rearranging mutations. So we actually have our own individual mutation spectrums, which is driven by some of the genes that we carry, which is a kind of surprising finding. So there's two of the reasons why PRDM9 is the most interesting gene in the human genome. Here's the third. Now, this is not a human, for those of you who are still awake. It is actually a mouse. And the observation, which is nothing to do with me, but is absolutely fascinating, is that mice have different species. In fact, they have different subspecies. And some of these subspecies actually can't reliably mate with each other and produce viable offspring. They're in the early stages of speciation. Now, people have been working hard to try and understand the genetics of that speciation, and you can guess which gene turns out to be critical to that speciation process. It's PRDM9, and indeed differences in this binding of PRDM9 between these different species. They've got PRDM9s going off like a rocket in these different subspecies, and when you put the two together, for some reason it doesn't work. And that actually seems to be important in driving speciation in mice. We have no idea if that's true for humans, whether it's, for example, true in the early human chimpanzee divergence, but it's obviously something that's fascinating, and in the next few years, hopefully, we'll learn a lot more about that process. Okay, so that's really all I want to say. Hopefully I've given you a couple of stories that give you a sense of just how bizarre the human genome is when you start to pull it apart in detail, how surprising some of these processes like mutation and recombination, which are so fundamental to the way in which species diversity evolves, they can really have some very unexpected turns and consequences. Now, as I said, I've been incredibly lucky in the last, in fact, all throughout my career in working with fantastic people. And so really, the science is a collective effort. I have many, many people to thank. So rather than listing them all individually, I just want to put up a big thank you. And for those of you who don't know, this is a stained glass window in Caius College in Cambridge, which was put up there in honor of Francis Crick, who was an honorary fellow there. So thank you to all of them, and of course, thank you to listening. And I'm happy to take any questions.
Very much for that, and Professor McVean has already asked for questions, and the hands are going up. Now, before we nominate the first lucky person, there are two roving microphones, so when you want to speak, put your hand up and then stand up so that the camera can catch you when you ask your question, and we'll try to be one question ahead of ourselves so that the microphones can be lined up. So we'll perhaps take two from here, and then let's have a hand up over there somewhere. Okay, there's one there for the next one. Third one.
Let me see if I get the gene name right. I'm curious about the selective pressure for diversity in PRDM9. Is there any advantage to having this diversity, or any theories about why that's the case?
So, of course, it's something we think about. So we have a bunch of hypotheses. So there's one explanation which we at least have some of the evidence for. It's quite a complicated answer, so you'll have to bear with me. So there's this motif. PRDM9 comes and binds to the motif. And that causes a double-strand break, causes the DNA to break, and that's the way in which recombination is initiated. Now, you imagine a mutation occurring in one of these motifs. Okay? So an individual now turns out to have one copy of the gene with the motif and one that doesn't have the motif. Now, what's PRDM9 going to do? It's going to come along and bind to the one that has the motif. It's going to break that DNA. Now, that cut is not clean. The cut's eaten back. Okay? So you end up removing a bit of the DNA, including the motif that's broken, and it gets repaired by the other chromosome. So what happens is that this version that broke gets repaired by the version that wouldn't break. And so what you have is a natural selection process, again, it's exactly equivalent to Darwinian selection, that drives these essentially self-destructive mutations through a population. Now, we actually have direct evidence that that's happening. You can see it. Actually, you can see it in sperm, but you can also watch it between humans and chimpanzees and see that it's happened. And so it's quite possible that you end up erasing your motifs. So you've got nowhere to bind, so you need to change in order to be able to bind somewhere else. So that's one hypothesis. Whether it's the full story, I doubt, but it's almost certainly part of the answer.
Second question here.
Going back to your story number one, you showed very elegantly how this paternal age effect of mutations was particularly strong for the kinds of mutations that enhanced cell division itself. But I was just wondering whether there isn't also evidence for a fairly noticeable paternal age effect as opposed to maternal age effect for mutations generally, because after all, the male germline is long and indeterminate. It's almost like a chemostat that's proliferating without natural selection away of mutations throughout life, whereas, as we know, the female one is very different. And you can.
Yes.
Is there evidence?
So you're absolutely right. So there is a paternal age effect for all types of mutation, almost certainly, all sort of replication-associated mutations. And one evidence for that is that if you compare rates of evolution on, say, the X chromosome, which spends most of its time in females, versus the rest of the genome, then you see the X has a much lower rate of evolution. And that's probably driven by this effect. And so estimates tend to say that the fraction of mutations that come from men is about 6 for every 1 mutation through females, generally. So it's a 6-fold increase. The types of things we were looking at there, the base rate, the increase over the sort of rate of point mutation is orders of magnitude. It's not 6, it's thousands. And so that's the sort of the big difference. Yeah, I mean, it's always bad being an older male, and for different reasons, basically.
I ask this question without a vested interest because I'm no longer especially concerned to reproduce.
Thank you. Super lecture. Thank you very much. Could I ask a question about the results of the 1,000 Human Genome program? Is there any evidence from it so far, either to support the alleged dramatic narrowing of the human population some tens of thousands of years ago, on the one hand, or for example, the subsequent and presumably more recent, but presumably minor variations in between human races that cause us to look each slightly different, one from another?
Yeah.
Thank you.
So the 1,000, for those who, most people won't know, the 1,000 Genomes Project is sequencing individuals from across the world in sort of particular populations. So far, we've only done a pilot study, but that pilot study did involve one set of samples from a European ancestry population, one from an African ancestry, and two separate East Asian populations. And so we can try and ask some of these questions. So in terms of your first question, did we see a big squeeze in diversity about 70,000 years ago in the sort of the peopling outside of Africa? Yes, I mean, that's in line with other studies using more limited data. We see pretty much the same kind of pattern. You know, what we found hasn't substantially changed the inference that about 70,000 years ago, when people left, when modern humans left Africa and peopled the rest of the world, there was a squeeze in diversity. There was a subsequent squeezes in diversity moving into East Asia as well, and then over into North America, there was even more squeeze. So that picture stayed fairly constant. One of the great things about sequencing individuals is that you can be completely comprehensive about the variation. And so we can really ask these questions like, do we see fixed differences between Europe and Africa? You know, how big a difference do we see between Europe and Africa? And the answer is, you know, it's actually very little. I think there are 2 places in our genome where we found mutations that are completely fixed for one version in Africa, completely fixed for another version in Europe, for example. One of those is associated with skin pigmentation. No big surprise. One of those is associated with your ability to resist Plasmodium vivax infection. So where selection has found a phenotype that it can act on, where a single change is massively beneficial, it can make these big differences. But in general, for the vast majority of our genome, the populations are very, very similar. You know, there's some sort of simple measure that people use to say how different are populations, or how much of the total variation is due amongst human genomes is due to differences between populations. The typical number that's banded around is about 10% of all the variation is due to differences between, say, Europe and Africa, and that's the kind of numbers that we're finding. What we are finding that I think is an interesting sort of corollary to that is that, you know, that's looking at the deep history, but we're now sort of looking, now in sort of subsequent phases of this project, we're looking in much more detail at very closely related populations. So, for example, individuals from Northern Europe and individuals from Italy, or individuals from Northern China, individuals from more southern parts of China, but who ostensibly would have essentially the same ancestry. And what we find is that generally they're identical, that you know, pretty much identical, particularly in this sort of how much variation you sort of is due to differences between these populations. But as you go more and more into the rare mutations, the things that are sort of 1% frequency in one population or 0.5% frequency, those are turning out to be quite substantially different. So what it looks like is that when you get down to very rare variation, populations can actually be quite different from each other. But that's not because, you know, it's not like this mutation that's fixed here that for one version and a different version over here. They're just collections of rare mutations that one population may have that another one doesn't. Okay.
Any further questions? No? Right. We're spot on time, actually.
I don't know if you want to.
Did you have another question? No. We promised a lecturer that if he spoke for an hour and if he sang for his supper, he would actually get fed, and that's what's going to happen. But before that, I'd like to thank Professor McVean for an absolutely splendid lecture, fascinating lecture by a real expert. I think it was very clear to understand, and we will all probably look at our neighbours slightly differently now, wondering how many mutations or not there are different. So thank you very much indeed for that. Very excellent choice for this award, I might say so.
Thank you.
And for his work to date and for his lecture this evening, he receives a scroll.
Thank you very much.
And I think this is the Francis Crick Medal.
Brilliant.
Award
Lovely.
medal.
Thank you.
And importantly, a cheque.
Thank you very much.